So we're at our next sample in our transect, and you can see there's a different composition of plants here making up the plant community. So we're going to go through the drill of identifying the species present and then deciding what percent cover class we think it belongs to. So there's a few different plants that you can see very clearly here. There's this pinkish plant that looks like it's taking up a lot of this area. That's called Polygonum pennsylvanicum.
Yeah.
Correct?
Okay.
Okay. Alright. So we would write that down. And then another one that you can see here is the Polygonum punctatum.
Correct.
Alright. So that's what a lot of this white color is coming from. But then in addition to that, we have a little bit, I'm trying to find one that's in it. Should I count this? More. I thought we saw some that were
in it. Yeah, there's a little of the. Oh, wait. He's not in there. Oh, he's not actually in it. Yeah, the terthum is not really. Oops, sorry. The terthum's not really in it. There he is. He's right next to it.
okay. So if it's next to it and kind of overhanging, we don't count it, I believe. Alright, and then the yellow flower you see here, is this a flower? No.
Yes.
Yes? Okay. Alright, this yellow flower you see here, this plant is the goldenrod that we saw at the previous transect, at the previous sample. So we've identified that one. And then we have this grass. You can see kind of below some of these greener plants. This looks a little dead. And we don't have that in our identification guide, but that's something we could take a picture of and then try to identify it later after we've recorded that we saw it. This is technically Transect 1, Sample 3. So we saw the Polygonum pennsylvanicum, the Polygonum punctatum, and then we would have to come up with a shorthand for the flat top goldenrod we saw because we don't have the species name on hand. So you probably want something shorter than what I wrote here. And then the unidentified grass that we found. So now we can figure out our percent cover.
Okay.
Alright, so I'm going to go in the same order that I wrote them down. The Polygonum pennsylvanicum, I would say it looks like it's got a good 50% would be my estimate. I can't.
Yes, I agree.
Yeah,
I think we're right at 50% with that one.
Yeah, it seems hard to discern if it's greater or less than 50%. So
Might be just a tad less.
the 7th cover class is 25 to 50, and then the 8th is 50 to 75. So we would have to use our best judgment.
Yeah, you could actually, when it looks like it's exactly 50%, you could put 50 in the.
Okay.
Which is an easy thing for the eye to discern. So it's perfectly acceptable to do that.
Okay. And then the Polygonum punctatum, there's just a little bit over here, but it's mostly in that area. So I would say that's probably over 25%, because it looks like in this area it's about a third of it. So I would say in between 25 to 50%.
Right, on the high end. That one looks like it's pretty close to 50% too, if you compress it all into one.
Yeah.
So.
Yeah, because we have 1, 2, 3, 4. Okay.
Right.
Because this is 50.
And
Yeah, but it is missing. Okay, should I write 50%?
yeah, I would write 50 for that one too.
And then the goldenrod, it doesn't really look like it's anywhere except for right here. And 25% would be this whole quarter of the quadrat. So I would say this would fall in 10 to 25%.
Yes. Yeah, probably closer to 10.
Yeah, so that's 6.
Right. So, and you can put a little down arrow to indicate it's at the low end of that spectrum. Okay.
Yeah, because I guess 10% would be 10 of these squares.
Right.
So, yeah, so that makes sense. You could argue that it could be closer to 10% and then... Okay. All right, and then the grass, we don't really have that much of, so I would say 1 to 2% would be 1 or 2 squares. So maybe because you can see it's kind of taking up 2 or 3 squares. So I would say either 1 to 2 or 2 to 5%. What would you say?
Yeah, I think 2 to 5 is good.
Okay.
Yeah, you come to some consensus on what it is. So number 4. Okay, awesome.
Yeah.
Okay, so that completes. There we go. How well you can see that.
And so
go ahead.
I was just gonna say, if you do just write the percent, making sure you have that unit there so that you know it's a percent rather than. I mean, it's kind of clear that 50 is not a cover class, but it's always good to have your.
Right. So we end up having 15 samples of the plant ID and percent cover because we have 2 30-meter transects and we go every couple meters to take our samples. And so you would have to take this back to the not to the lab. You could do it just on your laptop at home, and then you have to construct a spreadsheet with all the plant IDs and the percent covers in it. And we would take a picture of the grass, for example. Here's a good example of the grass. We would take a picture of it or take a physical sample. That would allow us to ID that plant and get the scientific name for it back in the lab. And it's the same with the flat top goldenrod. That one's a pretty easy one to find just by putting flat top goldenrod in. So we could get the scientific name.